You are welcome to this session. We are going to look at viruses. Viruses are also infectious agents that are much more smaller than bacteria, and they cannot produce on their own. They cannot reproduce on their own. So because they cannot reproduce on their own, they require a host cell to replicate and cause a range of diseases. Some of these diseases range from the common cold, which most of us suffer from, to AIDS. Viruses also have their characteristics. So let's look at the viral characteristics. One of their characteristics is that they are infectious agents. In other words, they cause disease, and for them they are much more smaller than bacteria, and for them to continue multiplying or reproducing, they require a host cell to reproduce. Remember also that viruses are not considered as living organisms. Why? Because they don't have the ability to reproduce or carry out metabolic activities on their own. Instead, they hijack the machinery of host cells to replicate and cause disease. Classification of viruses. We are going to classify viruses according to their genetic material, their shape, size, host range, disease type. That's how we are going to classify the viruses. Let's look at the size. Viruses can range from around 2,000 nanometers, for example, the parvoviruses, to over 400 nanometers, for example, the pox viruses. Let's look at classification by host range. Remember, viruses can infect different types of hosts. These can mean animals, plants, fungi, and also bacteria. Classification according to disease type. Viruses can cause respiratory diseases. That's why they are known as respiratory viruses. For example, the ones which cause influenza. We have gastroenteritis, which is caused by the norovirus. Then we have the sexually transmitted viruses. For example, we have the human papilloma virus. Let's look at the structure of the virus. The image depicts the structure of the virus, whereby the outermost cycle is known as the envelope, or a bubble of fat, followed by molecules of protein. Then we have the capsid. Then we have the internal part of the virus, which is made of DNA or RNA gene. Parts of the virus. Let's begin with the innermost part of the virus, which carries the genetic material. The genetic material of viruses contains DNA or RNA. DNA stands for deoxyribonucleic acid, and RNA, ribonucleic acid. Okay? So the DNA and the RNA, remember, these are the carriers of the genetic information. They bear the information, or they carry the information which is responsible for making new virus particles. Then we have the capsid. Capsid is made of a protein shell. It surrounds the genetic material, and it also protects the delicate genetic material, and also it helps the virus bind onto the host cell. We have the envelope. Some viruses have additional envelopes that surrounds the capsid. The envelope is got from, or is derived from, the host cell membrane, and the envelope helps the virus to enter and also exit or leave the host cells. When the virus enters and when time comes it wants to leave, then it uses the envelope. Then we have spike proteins. Spike proteins, these are found on the surfaces of the virus, and they help them to attach onto the host cell. Let's move on to look at pathogenic factors of viruses. Pathogenic factors of viruses. One of it is cell damage. Viruses, when they attack the host cell, they will obviously damage the cell. When they have damaged the cell, this can lead to cell death and also eventually death of the tissue. How does this occur? This can occur through mechanisms such as disrupting cellular functions, meaning that the cell's normal functions are not going to be carried out anymore, including apoptosis. Apoptosis means prolonged cell death or causing inflammation. We have the immune response, which is also another pathogenic factor of the virus. When the virus infects a host, the host's immune system builds up by trying to eliminate the what? The virus. The host can mean an animal, it can mean a plant, okay? Or the host could be a human being. So when the virus infects a host, the host's immune system is going also to respond by trying to eliminate the what? The virus. This response can damage the host tissues, particularly if it is too strong or prolonged. For example, some viruses can lead to autoimmune disorders whereby the body's immune system attacks the host cell. To put it in the simplest way, for example, when the viruses invade the host's immune system, then within the host's immune system, for example, let's say that there are soldiers who are now trying to what? To eliminate the virus. Okay, so these soldiers who are trying to eliminate the virus, this can be compared to the what? To the immune system. So when it responds, it can either damage the host's tissues. This occurs when the response takes too long or too prolonged, okay? And when it takes too long or too prolonged, this can lead to autoimmune disorders whereby the immune system attacks the host's cell. Mutation. Viruses can mutate rapidly. When the viruses mutate rapidly, this can allow them to invade the host's immune response or develop new modes of transmission. This can also lead to new strands that can cause more severe diseases. For example, in the COVID pandemic, remember there was a certain time whereby they were saying that there are new strands of COVID virus that have mutated, and they were saying that those strands were much more stronger than the old ones. So it meant that the virus had undergone what? Mutation, meaning that the virus had changed its genetic makeup. Let's look at the viral genome and the replication. The genome of the virus, remember we said, or the genetic makeup of the virus is made of DNA or RNA. DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acid. And this DNA or RNA, these are found in single strands or double strands. So the replication of viruses involves the production of new proteins and nucleic acids, which can then be assembled into new infectious particles. In other words, when the virus reproduces, it means that it is going to produce more viruses that are resembling the first initial virus. And these ones that have been formed now, they are going to form infectious particles that are also continuing to infect the host cell. Let's move on to viral replication strategies. Under viral replication strategies, viral replication can involve different strategies. Number one, we have the lytic replication. Number two, we have the lysogenic replication. Three, reverse transcription. 4, RNA replication. 5, DNA replication. 6, assembly and release. Let's look at lytic replication. Lytic replication means that there is rapid replication and the release of viral particles from infected host cells. Replication means making a copy of exactly of the same virus, making a copy of the virus. So lytic replication involves rapid replication and release of viral particles from infected host cells. So this process will also involve lysis of the infected cells, meaning that the infected cells are going to rupture. When they rupture, then there will be release of new viral particles to infect other cells of the host. Let's move on to the second strategy, the lysogenic replication. Lysogenic replication involves integration of the viral genome into the host cell. It means that the genetic makeup of the virus is now going to integrate into the host cell's DNA. So when it does so, it allows the virus to persist in the host cell for an extended period of time. The viral genome can remain dormant in the host cell's DNA, or it can be activated to undergo lytic replication. Reverse transcription is a replication strategy used by rotaviruses, whereby the viral DNA genome is reverse transcribed into the DNA. The viral DNA can then integrate into the host cell DNA, allowing the virus to persist in the host cell. Then we have RNA replication. RNA replication is a strategy used by the RNA viruses, whereby you have the viral RNA is replicated by a viral RNA-dependent polymerase. This is an enzyme. So in this process, this can involve the production of subgenomic RNA molecules, which are also used to produce viral proteins. DNA replication. DNA replication is a strategy also used by the DNA viruses, whereby the viral DNA genome is replicated using the host cell DNA replication machinery. So this process will lead to the production of intermediate RNA molecules, which are also used to produce viral proteins. Assembly and release. So once the viral replication is complete, then it means that the new viral particles must be assembled and released from the host cell. This process involves the packaging or the packing of the viral genome and the viral proteins into a new viral particle, which is then now released from the host cell through lysis or budding. Let's look at the viral life cycle. The viral life cycle is made of a series of steps as the virus undergoes to replicate and produce new infectious particles. There are three main stages of the viral life cycle. Stage number 1 is adsorption. Stage number 2, penetration, uncoating and replication. And stage number 3 is assembly and release. Let's look at the first step. The first step of the viral life cycle is known as adsorption, where the virus, or here, the virus attaches to a specific receptor on the host cell surface. After attaching on the host cell surface, then this is followed by penetration, whereby the virus will enter the host cell. Some viruses can enter the cell through a process known as endocytosis, whereby the virus is engulfed or eaten by the host cell membrane, while others can fuse with the host cell directly and release their genetic material into the cell. Uncoating and replication, the second process. After the viruses have entered the host cell, it undergoes uncoating, whereby the viral nucleic acid is released from the protein coat, or it is removing its coat. Put it maybe that way in simple terms, okay? So after the virus enters the host coat, it undergoes uncoating, whereby the viral nucleic acid is released from the protein coat. The virus then uses the host's protein machinery to replicate its genetic material and produce new viral proteins. This process can also involve different strategies depending on the type of viruses, such as the production of RNA from a DNA template, which occurs in the retroviruses, or RNA from an RNA template, which occurs in RNA virus. Assembly and release, this is the final or the third stage, And this is where the newly produced viral nucleic acid and proteins are assembled into new viral particles. These viral particles then exit the host cell through lysis, whereby the cell membrane ruptures and the virus is released, or budding, whereby the virus is released from the host cell membrane, and this takes place in the host cell membrane within it. So it means that the newly formed infectious particles can now infect the other hosts. Let's look at serological and molecular diagnosis of viral infections. How shall we diagnose viral infections? Serological assays involve detecting the presence of virus-specific antibodies in the patient's blood or in the bodily fluids, which can indicate a current or past infection. Then we have molecular diagnostic techniques. For example, PCR. This can be used to detect the presence of genetic material in patient samples, and this usually provides a more sensitive and specific method of diagnosis. Prevention and treatment of viral infections often involves health measures. For example, vaccination, quarantine, whereby some of the people or the individual who is having the disease is isolated, and also contact tracing. Those people who have ever suffered from the disease, they try and look for them so that they can be isolated. Another way of prevention is by use of antiviral medicines. These can be used to treat certain viral infections, but at times the effectiveness of treatment will depend on the virus and also the stage of infection. Vaccines. Vaccines are very important in prevention and control of viral infections. And how do vaccines work? Vaccines will stimulate the body to produce a protective immune response against the viruses. So you now find that the development of vaccines involves identification of viral agents that will put forward an immune reaction and creating a vaccine formulation that can safely deliver these antigens into the host. In other words, when they are preparing the vaccine, they are trying to prepare viral antigens, and these viral antigens will stimulate the body to develop antibodies against that particular virus. Emerging viruses have recently appeared in the population or in the geographic area, and this has increased a lot of what? This has increased incidence of severity. For example, the COVID-19 pandemic, which was caused by the SARS-CoV-2 virus. Factors contributing to emergence of viral diseases include the following. One of the factors include environmental factors, increased travel and trade. Human beings keep moving from one country to another because they have to trade, of course, and they need to survive. But in between, then there is carrying of these viruses from one country to another, and also changes in human behavior. Many emerging viruses are zoonotic, meaning that they originate in the animals, then later they are transmitted to humans. Spillover events: at times a virus jumps from an animal host to a human host, and this occurs through various means. For example, when you are in direct contact with these animals, most especially people who are used to hunting the wild animals and they eat them, for example, the monkeys. Consumption of contaminated food, meaning that the animal maybe that has been slaughtered could be having the virus, or exposure to environmental sources. Some of the zoonotic viruses include the following. We have the Ebola, the SARS, and the HIV that have caused a significant public health hazard. Thank you for listening. Have a blessed day. See you next time.